Now coming towards IgE. It possesses the standard structure of an antibody with 2 heavy chains and 2 light chains. IgE is medically important for 2 main reasons. First of all, it mediates hypersensitivity type 1 reactions during the immediate phase, and it participates in host defenses against certain parasites such as helminths. Although IgE is present in trace amounts in normal serum, people with allergic reactivity have greatly increased amounts. Especially during type 1 hypersensitivity reactions, the Fc region of IgE binds to the surface of mast cells and basophils. This bound IgE serves as a receptor for antigen or the allergen. When the antigen binding sites of the adjacent IgE are cross-linked by these allergens, immediate degranulation occurs, and several mediators are then released by the cells, such as histamine, tryptase, and leukotrienes. This process is usually involved in everyday allergic reactions, such as hay fever, eczema, and urticaria. But they may also be observed in the rare and terrifying anaphylactic reactions, especially to bee stings, peanuts, or during allergic reactions to penicillin. Now, with the systemic release of IgE, there is bronchospasm, vomiting, skin rashes, edema of the nose and the throat, and vascular collapse may also be observed, which is sometimes fatal, while a more localized release depends on the site of exposure to the antigen. The type 1 hypersensitivity reactions also underlies many cases of asthma, where the constant triggering of the local inflammation leads to the hypersensitivity of the lung wall and the subsequent prolonged bronchoconstriction and airway obstruction. Recently, a humanized monoclonal antibody against IgE has been approved for the treatment of severe allergic asthma. This IgG antibody efficiently removes the antigen, and so it can protect against the mast cell degranulation. IgE is also the main host defense against certain important helminth infections, such as strongyloids, trichinella, ascaris, and the hookworms Necator and ancylostoma. The serum IgE level is usually increased in these infections. Because the worms are too large to be ingested by the phagocytes, they are actually killed by eosinophils that release worm-destroying enzymes. And lastly, we have immunoglobulin IgD. It has no known antibody function, but may function as an antigen receptor. It is present on the surface of many B-lymphocytes, and it is present in small amounts in the serum. Okay, so now that we have come to the end of our discussion about the immunoglobulins, let's summarize the main features of it. The immunoglobulins are classified into IgG, IgM, IgA, IgT, and IgE. The IgG, IgD, and IgE exist as monomers, while the IgA is a dimer and IgM is a pentamer. The IgG is responsible for the secondary response, type 3 hypersensitivity reactions, and opsonization, whereas IgM is produced early in the primary response, is responsible for agglutination and complement fixation. Coming to IgA, it is produced in body secretions that function as a protective barrier. The IgD is not known to have much clinical significance. And IgE is significant for type 1 hypersensitivity and host defense against helminths. Hopefully, we have answered all your questions related to immunoglobulins, their structure, and their functions. If you have any other queries or any other questions, do not hesitate to comment down below. We are here to help you. And do not forget to stay tuned to sqadia.com. We have many more exciting videos coming for you.